to our virtual comparative genomics workshop. The Institute of Food and Agricultural Sciences, the University of Florida, and the National Science Foundation sponsor our research group to give 2 or 3 days workshops covering this material at the University of Florida and other institutions. IFAS, UF, and NSF have also sponsored the creation of this series of video lectures. This is the 5th in our series of videos, and it is entitled Trees and Subsystems in Seed. The material covered in this lecture was prepared by Drs. Andrew Hanson, Valerie de Crécy-Lagarde, and Svetlana Gerdes. Drs. Hanson and de Crécy-Lagarde are both professors at the University of Florida. Dr. Gerdes is a member of the Fellowship for Interpretation of Genomes. She is a highly experienced genome annotator, well-versed in metabolism and familiar with the seed. The introduction, audiovisual material production, and editing of this series of lectures is done by Dr. Claudia Lerma Ortiz. We strongly recommend you to study the first part of our Introduction to Seed, which is in our 4th lecture, before proceeding to watch the material in this video. We begin our 5th lecture by explaining the anatomy of the trees tool in seed and how to use this device to find gene fusions. Then, we give you instructions on how to enter the seed platform starting from a gene name or via the subsystems. We do a further examination of the Compare Regions tool and its utility, including other methods to determine gene clustering, doing clustral protein alignments, and getting neighbor joining trees. We also teach you how to extract valuable data not only from the subsystem information page per se, but also from the subsystems organization by analyzing their functional roles tables and applying coloring and limit display tools to their spreadsheet tables. Finally, we describe in detail the practical value of the visual aids and links given in the feature evidence tools, including functional coupling analysis, fusion discovery, and literature references. If you need more information about the seed, please read the following paper by Rose Overbeek et al. 2005. The Subsystems Approach to Genome Annotation and its Use in the Project to Annotate 1000 Genomes, which you can get in the link below. Or go to the Seed Platform's main webpage. We will now proceed with our lecture. We recommend using Firefox as your web browser. We will go directly where we were when we finished the first part of the Introduction to Seed in our 4th lecture. You can do so through the web address shown below. This link will take you to the annotation overview page of GTP cyclohydrolase 1 of Propionibacterium acnes. We will also need you to open our lab webpage in a new tab with the address given below. Full instructions for our comparative genomics workshop are written on this webpage. To follow this particular lecture, please scroll down to the 2nd theme of the page where we explain how to enter seed using a protein sequence. Please scroll down to the Trees Tool section of the theme. Let's start now.
Okay, so we're all on this page. Yeah. Now, there's a little link here which says alignments and trees. Click on trees. And what you will see is a series of choices of tree. They're often not very different. In this case, I really doubt that they are, because the count, which is the number of proteins involved, is very similar in all of them. So just click the first one and then go update. So what seed contains are pre-computed phylogenetic trees for your gene of interest, and they are very interestingly colored, which enables you to infer quite a lot about them. Anything that has the same annotation will be given the same color. So you can look down here and see a whole bunch of Burkhold areas have been grouped together in a clade. You'll see that they have little action boxes in front of them. What the action boxes do for the moment doesn't matter. What I want you to look at is the color of the action boxes, because these are very cleverly developed. The Arabidopsis one is a good one to do, but this is the most obvious one. The colors mean the following. The colors around these action radio buttons and boxes are conceptually as if the protein is stuck on a spectrum and you are looking at light that hasn't been blocked. So if there's really no color at all, these proteins are sitting on top of each other. If there's blue, is saying there's something stuck on the N-terminus of your protein. And conversely, if you get red color, it means something stuck on the C-terminus of your protein. So if you see color here, not only does and the intensity of the color is some measure of how much is sticking out.
Of the length. So
yeah.
Often you have a light color, it's not really meaningful, it just means that the gene is a bit bigger. What you have to look is really the bright colors. The light colors usually is just differences in starting.
So these have already been annotated as it's a fusion gene. It's actually our friend from the Lactococcus group, but it's a fusion gene between two folate synthesis enzymes, or fusion protein. But if you didn't know that, if this was only annotated GTP cyclohydrolase and you saw a color like that, what you would do is click on one of the things there and you would go to its annotation overview page, and then you can click the CDD link, and then it will show you what the domains are. And as you expect, it's got the FOLI domain, and you see on the N-terminus this HPPK FOLK domain. So bright colors, red or blue, signify a fusion. Now sometimes those fusions will have been correctly annotated, but the ones you're interested in are most likely the ones that haven't, because by definition, if you're a researcher, you're looking for things that aren't already known, and so it will be proteins that show up with bright colors in these boxes which may have additional functional domains that haven't been annotated. So here we are back to our tree. You might just for interest, go up to Methylobacterium, which is sitting about halfway down the page here. Go to that one and click on that just to see what it looks like. So there you go. This does indeed have some kind of N-terminal extension, but this might be spurious. It might just be, it's not a conserved domain. So it's either something special to that organism, or it's possibly a miscalled start site. But again, the tool has done its job. It's told you, it's flagged a protein that it was blue colored, so it means there's something on the N-terminus which isn't on the other proteins in the collection. And because fusions between proteins are really convincing evidence of some kind of related function, fusions are a very rich source of discovery. So tools that help you to find them are very valuable. So that's the end of that first exercise where we entered from a protein. We're now going to do a couple of other things. We went into seed the first time from a sequence using the GTP cyclohydrolase 1 from plants. You can also go in just like you can with the string with a gene name. So we're going to do that now. So I want you to go back out, close the tree, close the peg page, and just go back to the entry page of the fig page, which is this blue thing with the boxes on it. We want the University of Chicago. Are we all ready? Okay. Now, we're going to go in with a gene name, and we're going to go in with this name of the gene that until recently in Lactococcus was not known to be what it is, which is now known to be this folate synthesis gene FOLQ. Its systematic name, you might remember, is YLGG. So to do this, you go to the box that says search pattern. Let me get it in the middle of the screen. In search pattern here, put YLGG. Doesn't matter lowercase or uppercase. YLGG. And now we're going to pick a genome from the box just like we did before, and we're going to pick Lactococcus lactis, subspecies lactis. Remember that subspecies lactis is important. So Lactococcus lactis, subspecies lactis. And then when you've highlighted that, you push the button search genome selected below. So the way I use this tool as a non-E. coli person, I mean, if I've got an E. coli gene name and want to find the protein, I put it in there and I go to the E. coli gene. And you can do that for you can do it for yeast, you can do it for Arabidopsis, you can do it for all the model organisms. So we get this page which gives you a link to what we were calling the Facebook page of that gene in Arabidopsis. So that was just a different way to get to this page for a gene via a name, either a systematic name, or these anonymous YLG whatever names, or you can get in via an official name like Folly or PyD. So now we're going to further explore this wonderful compare regions tool a little bit. First, I want you to click on tabular region information. The default is visual region information. Click on tabular and scroll down, and you will see that our gene, the one that we went in with, this YLGG dihydroneopterin triphosphate pyrophosphatase, that's highlighted red, just as in the pictorial display the gene is coloured red, the arrow is red, and these are the flanking genes for it. Now, one feature of this is this button which says cluster. These are pre-computed instances in which the gene frequently clusters with something else. Yeah. So this is.
It does the analysis of your compare region where you go around.
Yeah.
It does it automatically and gives you the strongest clustering genes.
So yeah. So objectively rather than subjectively, it will tell you what's this gene strongly clustered with across all genomes. Now, actually, if we click on this, it won't give us anything, or at least I don't think it gives us much. If we click on it for this gene, how do you interpret that? I always interpret that as not much.
Yeah, that's not much.
Not much, okay. So that's we do that as an example of not much. But wait, there is something later on. If you scroll up to one of the flanking genes, which is a very important gene in folate metabolism, dihydrofolate reductase, and if you click the cluster for that one, and now you need to learn a little thing about this. Quite a lot of the displays in seed do this. They'll default to showing you a certain number of the total. So all you need to do to see the whole lot, which is what you want to see, is put the highest number there in that box.
That's to minimize to speed up your browsing. If you're just browsing, 10 is enough just to skim things.
So if you want to see them all, put them all, hit carriage return. And now we're seeing a cluster here between dihydrofolate reductase and thymidylate synthase. Very unusually, thymidylate synthase uses a folate as a methyl donor, and in so doing it uses the folate as an electron as well as a methyl donor, and so it oxidizes the folate from its normal tetrahydrofolate level to dihydrofolate. And dihydrofolate is the enzyme that regenerates the normal tetrahydrofolate. So these two things are in lockstep. This one has to have that to regenerate the cofactor. So it's an example of a cluster which is instantly logically understandable.
Do we assume that these whole tables will be populated eventually the more information is known?
I think yes. In general, as the number of genomes go into seed and as they work through computationally the clustering, these tables get more elaborate. I've seen them do that. I don't think they'll ever be all populated completely because some genes just aren't that strongly associated. Remember I told you this morning about this estimate I've seen is about 40% of bacterial genes are clustered. In some organisms it's more than that, in others it's less. Cyanobacteria it's less as a general rule. So you could perhaps expect eventually that maybe something like 40% of these things would show up as being in conserved clusters.
And so the different entries are all from different organisms? The different rows, they all come from the same organism and they're different clusters from the same organism, and only in that one cluster the genes are found? Or what does it actually mean, that table?
Okay, so seed was, it's a distributed system. So if you wanted to, you can install a version of this on your own university and load only the genomes you want, only subsystems you want. And we did it so that maybe industry would use it and university would use it, but we still maintain several seeds we maintain centrally, and then everybody else can take their seed and go. Since it's impossible to maintain so many seeds, so we maintain a central system, which is called sapling. And it has all the genomes and all subsystems. But every seed only displays from that huge universe the chunk that it's geared to display. So it means that in this seed, which is a practice seed, a sandbox, only Streptococcus gardoni is fully present as a genome with all this underlying data, pre-computed similarities, functional coupling. Every other peg does occur in some other instance of seed. It's just not fully present in this instance of seed. It's technically, if you click on this peg, try it.
Peg am I clicking?
Any peg that has nothing, and it would say it's an arrow.
Yeah.
See, but it's not an arrow. It means in this seed you're in, which is a small, tiny seed, kept small for the speed of tutorials and stuff. We're not maintaining every genome.
So there
But
are
on some other bigger seeds, this particular peg will have similarities, will have functional coupling. So this is our logo. It's not a bug, it's a feature.
So my answer was that we're not going to analyze this deeply. We're just going to see if there's anything there, we run with it, and if there isn't, we don't worry. That's the technical reason. Okay, so come back out of here, just back out to here, and instead of clicking on tabular region, click on sequences. So you just have to go back one and then click on sequences. So the default is DNA, but I would change it over to protein, and then you can click Align Sequences, and the first thing it will do is do a clusteral alignment of the sequences that showed up. Now remember, we only had 5 sequences in this display. If we changed it to 50 or 100, it would be aligning all 50 or 100. We're not going to do that because it's going to take too long, but it's a tool that's completely capable of handling however many sequences or however many genomes you put in your compare regions. Default is no tree. If you go across to neighbor joining tree and reload it, it will then draw you a phylogenetic tree of those sequences. And you can see right there that 2 of them are annotated as we've been doing, dihydroneopterin triphosphate pyrophosphatase. This one is annotated probable, and these 2 are annotated something completely different, but they're not in the same clade as those 3. So you see, this is quite helpful if you're trying to annotate the genes in a family. You can use this tool, and you know that some genes are real and that you've annotated those carefully and you've got good evidence that that's what they are. You could legitimately re-annotate that one as being this, because it's grouped with it. You might not want to do that yet with these because these are not in the same clade. And we won't do it yet, but you see these radio buttons and squares. These are things that you can use to take this annotation and transfer it to here. So there's annotation tools scattered all through the seed, and this is one of the places you can do it from these phylogenetic trees. Other features, if you go back out of here, right back out to the back to the protein table. If you want to download the sequences here in your, again, that you've stacked up in your compare regions, this could be 5, it could be 50, it could be 100. If you want to download them, you just click on that button and it will allow you to open a FASTA file in whatever you want to open it in. So you can just quickly get all the sequences that you've stacked up in your compare region tool in a FASTA file and then transfer them to some other program for analysis. And there's a couple of other points in the seed where you can do that. You can grab all the sequences in one of these subsystem spreadsheets, for example, and do it.
This is very useful compared to other tools because you can choose on your compare regions the ones that are only with the folate genes and not the ones that are with another pathway. So if you have an enzyme that has paralogs, you can decide to really look at the sequences of only the ones you're sure are involved in folate and not involved in another pathway. And to combine the compare region with grabbing the sequences, the one you choose, is very powerful.
So we've gone into seed with a sequence, we've gone in with a gene name. Now we're going to go in via a subsystem. So you'll generate a lot of windows in the seed. I want you to close all the windows except that entry page. As you start to work, windows proliferate and it gets quite difficult to troubleshoot what's happening, and there's actually some bad things you can do by mistake if you have too many windows open. So a good discipline is to close things you don't need anymore. So if you want to view subsystems, there's a little link on the front page here. It says Use the new subsystem editor. So open that in a new tab, please, not in the same window, in a tab. And this lists, and there's a long list of subsystems which mostly, these are a mixture of these, what I called this morning core subsystems, which are the ones for, say, folate biosynthesis or fermentative metabolism or tryptophan biosynthesis or histidine degradation, really universal core pathways that are well known. And then there are experimental ones. Well, we're going to go to a very well developed mature subsystem, which is Valerie's folate subsystem. So to find that, you can find it with your search tool in the browser, but you can also, in the subsystem name box, just type folate and then hard carriage return, and you'll get all the subsystems names which have folate in them. So if, for example, you want histidine synthesis or degradation, you put histidine in there and up would come synthesis and degradation. So now click on folate biosynthesis. And the first thing it does is open you to what's called the subsystem information page. And this has two sections, description and notes. Those are a bit overlapping in what people put in them. But typically in the description, there's a short summary of the pathway or the system involved, comments about genes that are missing, and notes might be a number of things. It might include, depending on the person who constructed the system, it might include outstanding problems, or as in the case of Valerie's subsystem, it's notes on variant codes, which we'll come to tomorrow. That's the face page for the subsystem. Click on functional roles now. This is the entry point. This is where you would put in the genes that are in the subsystem. So this is defining the columns in the subsystem, the enzymes involved and the transporters involved in a pathway. So you see Valerie's put them roughly in order in which they occur in the pathway. It starts with this one, it goes to that one, and so forth. And then you come down to things which are peripheral to the pathway, and there's altogether about 35 genes in here. This is quite a sizable subsystem. So this is where, if you were building a subsystem, and that's what we will do, you would add your roles. You build this table.
So can you look at the pathway somewhere as you're building it?
Some subsystems have exactly what you asked. Under diagrams and illustrations, they'll have a picture.
Oh, here's one on the illustrations.
Yep.
Here's ours. This is our baby.
So, and actually this is a typical seed format pathway where the intermediates are circles or ovals, the enzymes are boxes, and the arrows linking them are the reactions. I think Andre Osterman's systems are particularly nice,
Yeah.
and Dimitri's.
Not all subsystems have this.
No, not all of them have that, but thank you for asking that, because it's well worth clicking on the link to see if one is there. If it's there, it will be helpful. So, if we come back out of here, now we're going to go to the spreadsheet, which is the whole thing, which may take a little while to load. This is the spreadsheet where the columns are the functions, the rows are the genomes, and it will tell you at the top of the spreadsheet that there are actually 954 genomes in this, which must be pretty much the sum total of what's in this, in the Chicago seed. And these are the genomes, and there's 954 of them. They're arranged somewhat in taxonomic order, beginning with archaea, going through the different major groups of bacteria, finishing up with the relatively small number of eukaryotes that are there. I don't know how many eukaryotes are in this one. Oh no, sea squirt, chicken, everything is there. And the default, when you open these spreadsheets, the default is to have color coming on certain genes, and this is one of the most beautiful parts of the way the seed works. The color is a way of designating, showing clustering on the chromosome. So when you see that these 4 genes have the same color highlight, it means that in this genome these 4 genes are close together on the chromosome, and you can actually tell that by looking at the numbers, which are generally sequential. They're 512, 58, and 59.
Just a note, it's not always colored. It's colored here because the last iteration of this was colored. If you don't see any color, Andrew can show you how
Well,
to
yeah,
color.
we'll be going to
It's
the...
not always colored by default.
No, well, when we build our own subsystem, we're
Yeah.
going to color it, but yeah, that's important. It generally comes up colored, doesn't it?
Because you're looking at those that have been colored recently. If you build a scratch, or if you have uncolored it, it won't be colored.
Now, as my instructors in the army used to say, those of you who are awake will now have noticed that there are different colors. This is, again, the power of the seed, because these genes are clustered together, the red ones, the blue are clustered together, the yellow are clustered together, and the green are clustered together. So it's a way of showing multiple kinds of clusterings within a genome just at a glance. It's really brilliant. And as you scroll down here, you can see that sometimes there's a lot of clustering in the folate pathway. It's good in that regard. It's quite typical to have 2 or 3 or 4 different clusters with one block of the pathway in one cluster, or probably operon, another branch of the pathway in another cluster, and so on. So if you wish to see less than the whole spreadsheet, there's various ways to do that. For example, if you really only wanted to see the firmicute bacteria, you would go to the tab at the top, Limit Display. We're going to be exploring all these tabs in the next 2 days. It's a lot to do, so we're doing it stepwise. Just go to the Limit Display, and it says here, Limit Display Genomes. So if you wanted, you could scroll down there, and you could say, let's just see the firmicutes. That's a group of gram-positive bacteria. It includes our friend Lactococcus. And so it'll just show you the firmicute genomes.
genes. Those are actually pre-grouped. It makes, yeah.
And again, looking like what we saw this morning, you can see that these particular lactobacilli are missing great chunks of the pathway. So these would be examples of organisms that would pretty certainly need folate in their culture medium, and of course that's really not surprising because lactobacilli, they're called that because they grow in milk, and milk has folate in it for the infant, so they're in a very folate rich environment.
So in this table, it doesn't mean if it's empty, it's not a good annotation. It means they probably won't have it.
If a box is empty, it could well mean that the gene hasn't been given the correct name and therefore hasn't been imported into the spreadsheet. But in these mature core systems where a lot of work's been done, and if it's in multiple genomes and it has a pattern, it generally would mean it was missing. But there are ways to find that if it really is missing, and we'll be visiting those tomorrow.
That's the whole point of building a subsystem, is when you have a whole, is it a real whole or it's not?
Yeah. But with these subsystems are really resources for annotation of everything, and so these generally are correct. If they're missing here, they're generally missing. Now you'll see immediately that there isn't any color in this. This is because restricting the genome caused the color tool to go off. So now we're going to learn how to do what Valerie said, which is color the spreadsheet if it hasn't colored. You've got different options here. It's gone back to do not color. Color by cluster is what we were doing before. So color it by cluster, ask for it to be colored, and you'll get back just for the firmicutes now. And there it is, all colored again. Now, one or two other things you can do with this very easily. If you go back to limit display and display all genomes again, so you've got to put all, and then somewhat counterintuitively click limit display to limit it to all of them. So go back to this and limit display all. So when you've displayed all the genomes again, go back to color spreadsheet, and it's coloring them by cluster, but there's another way you can, there's several ways you can cluster, but the one I want to show you is where we're coloring not the clusters but genomes, and we're coloring them with respect to whether the genes in the genomes have been shown to be essential by systematic knockout studies, which are available for about a dozen total bacteria and yeast.
Still in the gene, so there are two ways to color the genes. So it means you color one cell at a time, or the genome, and then you color the whole row, right? So let's still stay in the genes.
Just watch me do it, and we'll just do it together with me doing it, because this tends to take a while. But most of the genomes in this 900 and some total are not going to be colored. Yeah, okay. So you click by attribute, and then you go to essential gene sets bacterial and color spreadsheet. And now it will go through and pick out of that 950, it will pick the 10 for which there are essentiality data, and it will color the genes on a kind of a color scale according to whether they were supposedly essential, supposedly non-essential, or possibly questionable, or not known, because the genes weren't knocked out in the study. And this generally takes, at the best it can be 30 seconds, at the worst it can be a lot longer than that. But it's worth seeing, because if you're working with a gene and you don't know, it's a novel gene, you don't know what it does, it can be very informative early on to find out, is it essential in any of the organisms in which it's been studied? And it's much easier to get it off the information from here than it is to go into the individual essentiality studies, of which about 3/4 of them are in this seed. Yes, Veda? Yeah, and there's a few that are not. So we'll give it a little while longer. If it doesn't want to work, we'll leave it working and look at it later. as I say, there's lots of things we can learn about these subsystems and how to extract information from them. Don't worry, it's coming over the next 2 days. We do it stepwise. Okay, so we've got the information. Red gene's essential, yellow gene's non-essential, blue we don't know because it wasn't knocked out. So, as I say, you won't see many genomes that are coloured, just the model organisms that have been studied. So here you've got Mycobacterium tuberculosis, pathogen, with GTP cyclohydrolase essential, dihydrogenoctinaldolase essential, Folk-A apparently not essential, which is a bit of a surprise. And then you go down again. Probably the gold standard knockout study is E. coli, would you say? That's real knockouts and real viability. So we'll go down, and actually you can always look for things in this system by using the search tool in the browser like that. And here's our E. coli with those being non-essential, that one being essential, and so on. So this is a nice way to compactly find a lot of information that would otherwise be difficult to get.
Now, Andrew showed you how to color by different attributes, for example, essentiality, and how to limit display. And I suggest to put them now together. So while you still have the spreadsheet colored by essentiality, go to limit display, and under user sets, locate the set of genomes that is said close to the bottom, essentiality data available. So remember, your coloring would still prevail, and you can put the tools. That's why we have so many boxes here and features that you can all mix and match, to some meaningful display.
So then if you want to color it with a limited display, it's quicker?
Right, it's quicker, and then if you want a picture for presentation, like Valerie said, you put time into making your display exactly as you want, and then you can, save, export data to Excel. So you can carve as I said, all the sequence space of microbes, color it to your liking, sort it, and then you export and publish or keep
We'll show
announcing.
you how to create a subselection of genome that you can ask for at any time. That's what she did. She selected some genomes, and now you can ask to see the spreadsheet only in those genomes. So if it's genomes you use all the time or a subset that you want.
Okay, so you should be
Just need to get everybody to Lactococcus lactis
yes,
foli.
exactly.
So
you have 900 rows in this table. 900 genomes are represented. It's very hard to find, a needle in this haystack. Two ways to do it. You can use your browser search. Just type Lactococcus lactis here, like Lactococcus lactis, and find it this way. Or if you can use the built-in filtering into the table itself. Right. So you can, again, as I said, so either your browser search or the built-in search into the system. And we're using the middle one. So please open the FOL-E gene in this genome, Lactococcus lactis, in a separate tab. FOL-E, it's the first one, conveniently. It's PEG number 1188 into Lactococcus lactis, the very first column. And again, you find yourself on this dedicated protein page, gene/protein page, because the information pertaining to gene and to the protein is all here. And we would revisit the feature evidence collection of tools and data. It's called feature evidence because in seed we're maintaining protein encoding genes, which named like that, as Andrew explained. We maintain RNAs, so non-coding RNAs, and then they would have RNA prefix here, as you may guess. And we maintain some regulators. So mostly in this tutorial, we would only be looking at PEGs, protein encoding genes, but you would not be surprised if there would be RNAs or any other abbreviations here. And those are all features. That's why we couldn't just call, protein similarities. So we had to, keep this naming very vague and open to RNAs, proteins, regulators, so we didn't really know how to name it. But for now, it's mostly collections of domain and sequence-derived information about this particular
So you can
protein
switch
encoding
feature
gene.
while you're working.
You can look
No,
at
you can, feature is a feature, so it's either protein encoding gene, or it's RNA, or it's a regulator. For example, okay, here you would see it's like proteins are arrows, RNA is just little rectangular boxes, regulators little squares, but they occur much more rarely than proteins, of course. It's not, I didn't want to do the sidetrack, I'm just explaining why the name is so weird, feature evidence, so that it would pertain to not only to proteins, to other objects you can encounter on a chromosome. So remember, it's a genome collection, genome database. It's not just protein. You can be, you're probably used to like PFAM and other collections, all they have is proteins. So we have the underlying DNA, we have RNAs, proteins, regulators. So it's everything that you can derive from a genome, more than proteins. But for now, we're looking at a protein encoding gene, which happens to encode 2 domains. So please, let's go again. Please follow along. Enter the feature evidence page, and it takes time to load because remember, these pages are so rich, and so the machine has to go to different depositories of data to bring it all in one display. And there are 2 tabs, visual protein evidence and tabular protein evidence. The heading would be the same. It just reminds you where you are in this dark forest. So this is the feature, that's the ID, from PEG, protein encoding gene, then the organism and the function. This just to remind you what your query is, where you arrived here. So please go to visual protein evidence. And we've already been here, but I just want to show more features. There's more information hidden if you hover. So try hovering over anything at all. See, the information is so multi-layered that if we would just type it all in, it would be a blur. So we're trying to follow Google's lead and just hide as much as you can, and just things appear on demand. But by hovering over organism name and the little bars representing the sequences, you can get a lot. So as Andrew said already, in every pair, the top line represents the query. So the query is the sequence you came here from. That's this particular Lactococcus lactis genome and this particular FOL-E, FOL-K fused gene. The bottom line is always the target, what the query hit in this genome of close relatives or more distant relatives as they go along. So the top is the query and the bottom is what it finds in this particular genomes. The color tells you how good the homology is, but there's more to that. So since you know a lot already about the top, the query, you came here from that page, but you're probably here because you want to see what are the homologs of this particular FOL-E, FOL-K bifunctional protein in other organisms. So the system tries to help you in that. So if you hover over any homolog, just for the hovering, you get the function, the subsystems, that particular homolog included in, how good the hit is. So it gives you how good the alignment is. But if you click once, it gives you a second floating window. As I say, we have to hide information in many layers. But if you click on it once, you get additional data. You get the name of this homolog, and this is the link. So if you click on it, you would be repositioned in the database to the homolog of this gene, but in a different genome. Do you see? It's not Lactococcus anymore. It's Deinococcus. That's how you can navigate this maze. Second, in addition to ID of the homolog found, you have all the subsystems that this homolog is associated with. And again, every name is an entry. Don't click it yet because it will take forever to open that subsystem. And on the very bottom, you can see the actual alignment. So if you want to see letter by letter, this little beautiful colored lines, just an overview. But if you want to go deeper, you can go deeper to subsystems, you can go deeper to alignment, you can go deeper to the protein pages. So Andrew already was here, but just repetition is the mother of learning. As Andrew suggests, and I agree, I would probably prefer E-value, blast score, over any other ways of sorting, but it's your preference. Please let us play with this group by genome. So ask for more homologs to display, and activate group by genome button and click resubmit. And the order of the homologs displayed would change. Now they appear in the order of how high homology is, but if you group by the genome, you would see that the homologs found in the same organism, for example, Thermoanaerobacter, they would be grouped together, even though the bottom one is rather yellowish and should be farther in the bottom. Its homology is not that good, it would have never been so high, but it was brought up here because it has a better homologue. And see how interesting, we know already that Lactococcus gene, that's why we're here, that's why we came. We knew it's interesting, it's a fusion, it has two domains. But interestingly, in other creatures, in this Thermoanarobacter, whatever is fused in the Lactococcus appears as separate gene. This gene has ID 2547, and this one 2545. So you can see they're next to each other. So in the genome of Thermoanaerobacter, just by looking at the IDs of the homologues, you can see that they are next to each other, 2545 and 2547. They're not completely adjacent, they're close, but they're separate genes, they're close, but somebody got in between. And this grouping by genome, by organism, can separate paralogs, or it can help you find the instances for homologues for every domain if they are not fused in other creatures. Now, this is of course the main attraction, this colorful display here, but there's more to this page. And for example, on the top of it, domain structure is shown. Again, if you just hover over it, it tells you first, if you just hover over, it's PFAM annotation of this domain. That's the first glance. And what's the score? With which score Lactococcus lactis gene is assigned in PFAM collection of domains. If you are interested in any of this, again, you would click lightly once and you would get another floating window that would actually take you there. Some links keep you inside seed. You just link, you can jump from one protein to another, from one organism to another, from subsystems to genes, from genes to roles, but some links are outside links. And again, at first it's confusing whether you are still in the seed or you're already outside. So this is outside, right. Localization only matters for eucs if there's information as to intracellular targeting of this particular protein. It would have been displayed here, but not always. I think that's good for now. Just note that this is the current functional annotation of not of the query, but of the hit, right? So the annotation of the query is on top, your starting point, and annotation of the homologues that are found in other creatures are displayed here on the right. And you can see the selection buttons. Keep repeating, seed is editable database. You can use the selection to select proteins to reannotate them or later, or you can select them to export and take to other tools or to your publications. For example, here, by clicking, you select. Again, remember, you select the homologue. You don't select the query, and then you can download the sequences or align them. And here you can or cannot include the query. By the query, remember, it's the protein you arrived here from, and it's a reminder here on the top, what is it, where you came from. Yet additional layer of information is hidden under the second tab. We've been here before, we just thought, by repetition, we can make you feel more at home. So this is the same information in a tabular format and with a few more added types of data. Again, the query here, not only it's here on the very top, it's repeated here. That's the query. It's on this brown highlighted color. And everything else below it is the same homologues in the same order that you saw them appear in the visual tab. However, here you have few more interesting things. First, again, a button to select for the same purpose, either to annotate, to export, or to get the sequence from. You have the names of those homologues. You have the detailed description of the strength of the homology. So this is how good the overall homology is. That's the percent identity. That's the region involved in alignment. So aligned position in the query sequence and aligned position in the target, in the hit. So, for example, to Lactococcus cremonius, out of 349 amino acids in the Lactococcus lactis protein, all of them are involved in the alignment, as well as in the target. So both sequences that are aligned, they're aligned almost perfectly. But if you go to the bottom of the sorted list, you would see that, for example, in the Streptococcus thermophilus, from the query, out of 349 amino acids of the query, only the first 133 is involved in the alignment, and they hit the target protein, the Streptococcus protein, they hit 133 letters out of 163. Do you follow? And if you're confused, you can click on this little icon of alignment and it would throw you back and try to position you, this is the streptococcus we were looking at. Okay, we can go here. Okay. And it's actually many ways to show the same thing. So these numbers, if you look carefully at them, try to convey which part of the query and which part of the target are actually aligned. And the color does the same. It's a spectrum. Now, to make the coloring even more complicated. So these 4 columns all try to describe the actual alignment. How good, 2 columns, and then what area of the query and the target are involved. And they have the coloring to convey that. This column with the names of organisms is completely separate coloring system, and that tells you the kingdom. This purplish means prokaryotes. Eucs are yellowish, archaea bluish. Right. What is important that the color in the alignment, 4 columns of alignment, has nothing to do with the color in the naming. So the color in this column only conveys the kingdom you're in, because that you would be warned if you're in Eucs already, or still in bacteria, or in archaea, or in viruses. So this column, the naming column, it's yet another system of coloring. The background color in the column with the annotation, with the name, only means that the annotations are the same. So the same color signals identity of annotation. So all GTP cyclohydrolase type 1 is all brownish. It means they're spelled the same, letter per letter they're identical, and they would go in the subsystem if you would ask the machine to associate every sequence with this particular annotation with your subsystem. I hope you follow. So the naming, we chose to highlight it with the color. I think we're the only database that does it, because names, in our case, is the only glue that puts the proteins into subsystems. It's very difficult for human eye to see a double space, right? But for machines, it's paramount. So we highlight with color the identity. So for whatever is on the white background, it means they're identical between themselves, and they are identical with the query. So the names on the white background are the same as the protein you came here from. And so it goes. For example, this purplish color, they identical between themselves, but different from the brown color, and certainly different from the query. And why do they different? Because the query has double name. It has this domain annotated and that domain annotated, right? But if a protein only has one domain, it would have a separate color. So we actually take a lot of pride in the fact that we annotate every functional domain separately. That helps you. The subsystem, the spreadsheet, it's very bookkeeping system, and it makes you organized. Even biologists like us, I mean, it makes us almost, mathematicians. You have the cells and you need to populate them. And if this protein encodes for 2 domains, well, to put it in 2 functional columns, we have to give it a double name. So this law and order that subsystem forces into biology actually pans out. So again, like in the visual protein evidence display, there's additional information. Similarities is where we just were, domains, identical proteins, and functional couple. Domains, if you click on it, it just takes you to the bottom of the page just to save you some scrolling time. And it shows you the same domains, but in a tabular format. Again, there's a visual tab, shows pretty much the same data graphically, and the tabular tab shows the same information in tables for those who like it more this way. But I wanted to show you because we didn't see it before. This is the table of this functionally coupled genes. So what this score, it's a very ad hoc, just devised score. It tries to convey how often the protein families occur in the immediate vicinity in the genomes of different creatures. So we started with this Lactococcus protein, and the score tries to tell us that at least in 23 taxa, 23 not genomes, but groups, its immediate neighbor within the 10 kb on the chromosome is the dihydropterate synthase. In a little less number of taxa, this particular protein family is its immediate neighbor. And with even less probability, you can find homoserine dehydrogenase. Again, these numbers, they are computationally derived and they're not current because SEED tries to keep current with the genomes that pouring in. It's like several hundred every month. So these numbers are already outdated, I can tell, but it just takes forever. And it's kind of a guidance. All it tries to convey is that with your query protein, these 3 protein families are very likely chromosomal neighbors. Doesn't matter with which numbers, they're just likely. So maybe they have something in common. And now, of course, they're beautiful annotated, and you can tell exactly why they're neighbors on the chromosome, and if you can't tell, ask Andrew. But in case that you start with a hypothetical protein, to know which protein families are statistically significantly close with it on the chromosome of different creatures without looking at these creatures is maybe valuable clues for the function.
The ones that end up here are really the strongest.
Right, they're
These are
pre-computed.
really usually
They don't depend on display, computed on the fly. This is
doesn't
rock
mean you
solid.
don't have others that are interesting,
Right.
but these ones are usually the top ones.
Right. Okay, just a few more things, if you can bear with me. So we only looked at the 4 columns that try to describe the alignment, then the organism, the functional annotation, and there are a few more columns which I really would like to mention. So associated subsystems is the same, the subsystems that your query protein is a member of. And as you see, each homolog has its own subsystems, associated with it. And as you go farther from the query, this list of subsystems can change substantially. So this list of subsystems in this particular display only tells you which subsystems this particular protein encoding gene is a member of. So it can be different for different homologs. For example, not every lactococcus genome has been associated with, Valery subsystem, for example. Then it wouldn't be listed here. Assign from and assign to are the ways that we actually edit and change functions, and we'll get later to that, so I wouldn't cover it now. But this I want you to know. So it's called evidence code, and when you have time, maybe not now, you can follow a little question mark and actually enter it and look. Oh, we can suppose do it. So as I said, we have 2 levels of hovering boxes. Just by hovering, you get 1 little overview information, but by clicking, you always get more, and sometimes 3 layers. You click once and then you click again. So you can try to read through that. But what we do here, I hope you still remember the string database that Andrew and you explored together earlier. They have different types of data that brings 2 little round balls, 2 different families together. It can be, literature mining, or it can be co-expression, or it can be conserved neighborhood, the genes that tend to occur in the immediate vicinity of each other in different organisms. So SEED values so much this clue, this how often the representatives of 2 protein families occur close on the chromosome, that we use that as a sole basis for this, what we call evidence codes. For example, ICW means in cluster with. It means this particular gene occurs in a cluster with, the number says with how many? With 2. In cluster with means not only it occurs close on the chromosome consistently in a conservative manner, it is also present in the same subsystem. So in cluster with is the strongest evidence we can give you of functional coupling. So it means that in this particular creature, this very peg in Lactococcus cremonius occurs in the same subsystem and is close on the chromosome with at least 2 other protein families. And you can explore them by going either into subsystem or into the chromosomal display, right? So it is the 2 non-homology based clues, we call them contexts, that we're trying to convey. So one context is genomic. So who are your neighbors? And not just neighbors, but conservative neighbors, and Andrew already described it very well. And the second context is functional. So if 2 protein families are the column in the same subsystem, if 2 protein families occur together in one subsystem, or 2 or 3, it also hints or screams that they are functional associated. So this in cluster with is a very strong clue that takes into account both. So there are protein families that A, occur in the same subsystem, and B, have representatives close on the chromosome. So again, now it doesn't matter, but if you work with your hypothetical and you see this ICW, it means good, you're on some interesting, functional clues to its functional and genomic neighbors. In subsystem unique means that this gene in all the subsystems it's associated with is only entry per cell. The flags to guide you to relevant literature are also in this column. It is not evidence code per se, but it's the way to find some experimental support to your function or your prediction. ILIT means indirect literature reference. It means the actual reference refers to a homologue of this particular gene. And DLIT, it means direct literature. Yes, for example here. DLIT means that this particular reference was generated for that particular organism. So the organism name is associated in PubMed with this instance of publication. But we thought that ILITs, so-called indirect literature, is also relevant. They help you if you are in a close homologue, just in a different strain of Bacillus. We thought it would be useful for you to know that somewhere for this protein family there were experimental publications.